Okay, so today we'll talk about Amplicon-based targeted gene metagenomics, and there are two kind of metagenomics analysis. One of them is Amplicon-based metagenomics data analysis. The second one is shotgun. And today we'll talk about this, and this is specifically about the bacterial communities. Okay, so there are many applications for this type of for sequence analysis. Um, so the idea about this amplicon-based metagenomics. So what we usually do, we take in the lab, of course, they take samples, and this sample taken usually from environmental samples. Environmental samples, that means from environment. What is environment? Can be from like human gut or human saliva, or can be taken from rumen, for example, or can be taken from the soil or from the air or from any environment. Then that will we can call that as environmental sample. So this is not like culture samples. In culture samples, we culture it in petri dish, in the lab. We culture it, right? But environmental samples, that it might include many microbes. So the purpose of this kind of experiment is to find the microbial communities. And we can use this in many applications. Some of these applications, those listed in this slide, are just part of this application, like microbial diversity in environmental studies. We may need to do, to do a study to make study like this, at least, to see how, like between two environment or two ecosystem or two samples, how microbial community are diverse. Okay. Um, and also you can use it like in human microbial studies. And this like, for example, if we take sample, we need to identify what kind of microbe in the sample, pathogen detection in clinical sample as well. We don't know exactly which bacteria, but by using this kind of analysis or this kind of experiment, then we will know the different kind of microbe in that sample. Also in food safety, we want to know the different the microbial communities, that mean all microbes in food. So to know this community, because some of them may be toxic, some of them may be pathogenic, some of them may be, just like they don't do anything, no harm. Okay. And in aquatic microbial ecology, also now it's become very important to know that, because also they do some kind of a study in aquatic ecology, like, in ocean and water, for example, like in Greensboro, they may want to know what kind of bacterial community in water or during the time. So they keep taking samples in time series, that means from time to time, and to see if this microbial community change or stay as it is. In case of, and also they can be using metagenomics bioenergy by identifying the methane producing archaea in aerobics um, uh, containers or something like that. They also use in evolution to study the ecosystem, because some bacteria, they will be reserved in ancient ecosystem, like, it's very cold weather, or they can be, just can be stored there for like million of years or thousand of years. Or so these will help to understand when you compare them with the recent species, then you will understand, the evolution and so on. And also in biomediation mediations as well for degrading what we call it polycyclic aromatics hydrocarbons, and those are they some material may have effect in human health. So sometimes we need to study this microbial community because some of these bacteria, they degrade these chemicals and make the environment healthy. So if like Pseudomonas, for example, and many bacteria, they can do that. And also with veterinary microbiome study in case in rumen to improve the uh, the nutrient, and in forensic microbiology as well. Also, they use it. So, this is just like examples of application, but uh, metagenomics, they have many applications right now, even in drug discovery also, and to discover also new antibiotics, or they can use it in that application. So the Amplicon-based, or by the way, we call it Amplicon-based or targeted gene. The reason why, because we use PCR, or polymerase chain reaction, as we discussed the PCR before. We said the PCR, the product of PCR, we call it Amplicon. Okay. Or also you can call it like targeted gene, because we target a specific gene. And in this kind of study, we are not using the entire genome, the whole genome, but we are focusing on a specific gene. And in case of bacteria, we are using 16S ribosomal gene. We can use any gene in the future if we find, that gene can give like, different, or help to classify the different microbial community, then we can use it. But so far, we know that the best gene that we can use in this kind of study is 16S ribosomal gene. And this is a DNA gene, but this gene can be a single RNA, and instead of forming bacteria, it becomes ribosomal RNA. So it will take part in 30S uh, ribosome units, and S is segmentation. So 16, this is just like the rate of segmentation. This is just a name. It is like 28, 16, 23, and so on. So if you ask about S, what does this come from? S means segmentation, because when you centrifuge the RNA, they will segment it in a specific rate. And so this 16 is just like unit of segmentation, okay? But we are using this 16 ribosomal RNA because can differentiate between the different types of bacteria. So why we use it, and we will show you the different part of this 16S ribosomal gene, but this is the one that we use in case of bacteria, prokaryote as general. And we said prokaryote can be bacteria or can be archaea. But if we need to do a study in fungi, for example, we can use 28, because fungi are eukaryote, they are not prokaryote. We can use 28 as ribosomal gene, okay? But for bacteria or an archaea, we're using 16 as ribosomal RNA. So this is something that I want you to remember. So we have zero data in the case of which contains the sequence. So here, the sequencing, it is high throughput sequence, right? That we get it from, high throughput technology. But it focuses in specific in this ribosomal 16S ribosomal gene. So there is no any other portion, and the protocol itself by focusing on. So if we need to focus on a specific part of gene or any part of DNA, that means we are using primer, right? Primer. You know this because this is just like a PCR. So this is the idea behind it. So here we are targeting a specific gene, and we don't focus on the whole genome. So this is the difference between amplicon-based and shotgun. So the bioinformatics analysis is used to analyze this data. So after um, the analysis, maybe we can get bacterial identification. That means we'll identify all bacteria in the samples. And also we can use what we call a diversity analysis. And there are two kinds of diversity analysis. One of them is alpha diversity, and the second one is beta diversity. We can construct value genetic tree um, for the various bacteria that will be in the community, and this will show you the relationship between the different bacteria, even they evolve or they are very similar or something like that. You will have idea about that. And also after that, you can do what we call it functional annotation. And functional annotation, it will tell you that if a specific number or a specific bacteria are present in that sample, they might be correlated to a specific kind of condition, whether that condition may be healthy, maybe something toxic, or maybe it's called diseases. But now they discovered that the bacterial community is really very important in health, and it can interact like in human. If a specific bacteria they are not there, human may be susceptible to some disease. And at the same time, if a specific bacteria that are there, in the environment of, also it may be susceptible to specific disease. So the presence of bacterial community really become very important because now they discover that it has some kind of interaction with human and with human health. Okay, because so this we say we focus on 16S ribosomal RNA gene. And when we say gene, that means this is a DNA. This is not an RNA. You should know that, okay? Because this is a gene that can be transcribed into RNA, and RNA will be part of the ribosomes. And if you see here in this uh, figure here, we have ribosome, considered as ribosome in bacteria. And ribosome consists of two units. One is 30S subunit, and the second one is 50S subunit. But in the 30S subunits, we have the ribosomes that actually this is produced by 16S ribosomal RNA. Then we have in 50S subunit, there are two other genes that code for these RNA. But we're not going to focus on this part, but we will focus on 16S ribosomal RNA because this we can use this to differentiate between bacteria. Okay. And 16S, it consists of course, it's like a sequence, right? It's like DNA sequence. And this DNA sequence can be divided into conserved region and hypervariable region. That means conserved region that the rate of mutation is very slow. The rate of mutation is very slow in conserved region. But in the variable region or hyper variable region, the mutation is very fast. So this one, and if you see here, we have some label V1, 2, V3, and so on. So if you they are interchanged with we'll find the conserved region, variable region, conserved region, variable region, and so on. So this is how this gene or this is the structure of this gene, how it look like. So that means we have both region. And this um, uh, composition make it unique and make it also suitable for this kind of analysis. Okay, because uh, there's like variable region and conserved region, variable region and conserved and so on. So here in the lab, what we need, we will the first step will prepare, we'll try to design a primer. We will try to design a primer like the when we discussed NCBI database, or we show you how you can design primer. So here, when we design any primer, we need to include part of the conserved region and part of the variable region. So we're not going to make it only from the conserved region, or and also we're not make it only from the variable region. So it must be this like it should span portion from the two part. And it can be from here, from here, from here, but there are many research that will advise you to focus when you design any primer to focus in a specific region. And if I can remember, it will be part of this part actually between portion of this region and V4. Okay. So before, just do this kind of experiment, you can hit some papers and try to see what is the best region that you can use it to design your primer. But um, I I I think most of the primaries, you can design primer in this region. And so when you use PCR, of course, we are using PCR once we have a primer. We are using PCR. That means we will amplify this region and we'll keep amplifying it. It will span, so it will extend. And it depends. Okay, it depends on the protocol and the primer that you use. You can maybe will uh, see, amplify all the gene or maybe just portion of the gene. Okay, so this is the how you do this in the lab. And once you do that, amplify that. After that, you send it to the sequencer. Then they will sequence um, they will sequence the sequence, and our expectation is the sequence the read that we will have in FASTA-Q file will include only the 16S ribosomal RNA and no other any other so so once we get the FASTA-Q file, and we know that the FASTA-Q file include 16S ribosomal RNA and no any other, that means a specific gene or a specific region of that gene. So our analysis, it will be straightforward. So here we adapt a specific analysis, and this analysis for amplicon-based metagenomics, we call it amplicon-based metagenomics. So here we have, these are the steps that can be followed. We have data acquisition. Of course, this is, you can get this either, from the lab, or you can get it from databases. We have databases that will find many studies. You can get, you can search in NCBI database and you will find, and you may find some something that may satisfy your study or what you are looking for. But most of the time you are using your own data. That means your own experiment that you design in the lab. Maybe you are taking sample, from water or from human gut or like, fecal sample or maybe whatever, something that you should. And always here, when we are dealing with something like that, always I want you to think about what kind of questions that you want to answer. Because the research itself is about answering question, right? Or solving problem. And this problem will be in the form of question. And what we do, it will be in the form of the solution or problem solving. So when we talk about something like that, that means we are addressing what we call experimental design. So you cannot approach uh, any metagenomic study without experimental design. That means already you have designed your experiment and you know what kind of question you are asking and what kind of answers that you are expecting. Okay. So in this case, maybe you want to compare between two uh, or the different groups, for example, the bacterial community in different group. What kind of this bacteria? Maybe like you have two lake and you take sample from here and sample from here, and you want to compare the microbial community between two and something like that. But anyway, um, this it will be determined by you. When we call it experimental design, that means you have to focus on experimental design before you approach any kind of analysis like this. But here, our part here, we have the data, the raw data in FASTA-Q file, and we know that the FASTA-Q file contain the ribosomal RNA only, that only RNA gene only, okay, so no other thing. The steps, then of course we have, so this is what we call it about data acquisition and pre-processing. Pre-processing, we'll come and discuss it, especially when we talk about the Amplicon-based, because sometimes maybe we'll do what we call it multiplexing. So we mix different multiple samples in a single run. So we call this multiplexing, and by pre-processing, we mean like, quality to make sure that the reads are of good quality. Otherwise, we will use any kind of mechanism that to improve the quality by trimming or filtering the read. And if they are multiplexing, we need to do what we call demultiplexing. That means we need to separate samples together. And by the way, multiplexing is a very good strategy because that it will lower the cost of sequencing. So if you have 100 samples, you can mix them all together in a single tube instead of running each one, right? But during the preparation, we need to annex or we need to add what we call it barcode to each sample. So if you have like 100 sample, for example, so before we mix them in a process of preparation, we need to add barcode to each of these samples in the separate tubes. After we make sure that these barcodes are added, that means each fragment will be connected to a barcode that unique sequence to identify that sample. Then, of course, we can mix them together. And after sequencing, that barcode also will be sequenced. So it will be very easy to separate the sample from one another. So if you have 100 samples, then we will have we can separate or read that with that specific barcodes, and we can put them in separate fast FASTQ file. So this is what we mean by demultiplexing and pre-processing. So once we finish this part, then we go to what we call it metagenomic features. And by here we have different strategies. We have two strategies. We can follow both of them or we can follow one of them. One of them is clustering, and we will come and explain how we do this clustering. And the second one is denoising. Okay, so it depends on you, and also it depends on your study. You can use any one of these methods, either clustering or denoising, or you can use both of them. That is fine. But the purpose of this sequence, the purpose of this step, you are getting what we call it features. And by features, we mean we are getting sequence. That sequence representing the microbe in that sample. Okay, if you have like different microbes, so we will get sequence that represent the microbes. And once we get the sequence after that, just of course we will identify the sequence and we'll know the taxa or the taxon of that bacteria, whatever. So feature here, we are using two methods or strategy, what when we call operation taxonomic units or OTUs. We'll come and explain this in details. The second one, feature, we are using amplicon sequence variants. Okay. So in case of clustering, we will get OTUs. In case of denoising, we get ASV or amplicon sequence variants. The only difference between is in case of OTUs, because this is generated by clustering, we don't get the exact sequence for bacteria, but we get representative sequence. But in case of amplicon sequence variant or ASV, we are getting exact sequence. So this is a difference. So here, if we need to be more specific, to be more accurate, so we can use denoising to get ASVs. Okay. So the second step will be taxonomic assignment. Of course, we have to identify because now we get the sequence or representative sequence, right? After getting the representative sequence, then we can use what we call taxonomy assignment or um, classification of taxonomy classification or identification of the bacterial community. You can use any name, okay? But the idea here, once we get the feature, then we can use, we have also many methods. We can use one of them to identify the bacteria or the microbe in our samples. We can go further after that. This is just one of the analysis. We can stop here, or we can go further if we need to construct a phylogenetic tree because we have sequence there, and we can use this sequence to see how these sequence related evolutionary from evolution perspective, how these relate to one another. And also we can use what we call it microbial diversity analysis. We have two kind of analysis. One we call it alpha, and the second is beta. Alpha, if we need to matrix, we need to find a matrix to measure the diversity of microbe in an individual sample. So here we have a sample, one sample, and inside this sample we have different bacteria, and we need a matrix. By matrix, something to measure the diversity in the diversity in that sample. It's just like we are all in the class. If we need to measure diversity, we need for metrics, just a figure, a number to tell us, how our class is diverse. This is the same thing here. We have sample, and in that sample we have many bacteria. So we need for a matrix that can measure the diversity in that sample. So are they like diverse bacteria, or bacteria are not diverse? They are very close to one another. And by better diversity, here, if you want to compare between diversity between two samples and more, so there are different indexes that or matrices uh, uh, that user can use it for the matrices. Sorry. So now let's talk about what clustering, how we do clustering, because we say there are two approaches. And please, I want you to focus on this, because these are the um, materials that I will bring in the exam and understanding them. Um, because I'm not going to give you any coding or stuff like that. So it will be about concept to understand this. We said there are two methods to get features. One of them is auto use, and the second one is ASV. Auto use only through clustering. And I believe, if you study, computational science or computer science, you may know what clustering is unsupervised machine learning. Okay. And if this like new term for you, it is a method that can group, if you have like different items, so you can cluster this group, so you can group them together based on a specific criteria. Okay. So by this clustering that we have we will get different sequences, and by clustering, that means we, based on similarity, and here we are using Hamming distant similarity, and I think we discussed this maybe in the alignment. We say that there are two different ways to measure the distance between two sequences. Hamming distance is one of them. So based on this, you can determine the similarity between any two sequences, right? So we can use clustering to cluster a group of sequence. You can put them into group based on similarity, and that similarity can be like 97, because there must be threshold for that. Can be 99. But when you get closer, that means also you may miss some information. Most of the time they use 97% similarity. Okay, because even if bacteria, there is some kind of diversity, right? There's some kind of variation. It is the same strain, or it is the same species, maybe also the same strain, but within the strain also you will find some kind of variation. You cannot find a bacteria like just human. You cannot find 100% identical gene. Okay, most of the time you'll find there's some kind of, but it's still that make it from the same from the same taxonomy group. So in this case, we need to cre to adopt a threshold or cutoff for that similarity. And most of the time, we are using 97% of similarity to cluster them together. That means if you have like many bacterial communities from different, they are very diverse. So we expect that if some sequence they have 97% of similarity, then we assume that they may be from the same taxon. Okay, they relate to the same species, genus, species, strain, and so on. Okay, so this is the assumption right here. If it's less than that, it might be from different group. So here you can be uh, uh, very conservative by increasing the similarity to 99, but also you are going to lose something because here you may get some kind of uh, like some already supposed to be related to this taxa, but because of similarity is very large because increase the number of similarities, so you may lose some of sequence. It might be classified to create, different cluster or something like that. So this is something that we need to look at when you are using these, but most of the time they are using 97% of similarity. So when we come to talk about the operational taxonomic units or OTUs, there are three ways to get it because this is clustering, as we said. It is um, unsupervised machine learning. Machine learning can be either, supervised or unsupervised. Supervised, like, decision tree or random forest, like is you support vector machine, like, there are many ways. Unsupervised machine learning, there is no label for class. We have just data, but the purpose here, we need to cluster them. And there is a different way, of course, of clustering with the hierarchy, there is a different way for clustering, so we don't need to discuss clustering itself because there are different kind of algorithm. Uh, but for using this, uh, for um, for using this method of clustering, there are three ways strategy. We have de novo clustering. That means here we are not depending on anything. We are clustering this from scratch. You are not depending. The only thing here we are depending on is the similarity. Okay. So we see, how the similar, which are similar, like 97 within 97%, if this is the threshold, we're going to put them in group. Okay. So we call this de novo clustering. So here we are not using any kind of database at all. We are depend solely on the similarity. Of course, some of these sequels they might not be similar, So they will be, just put in different group or when you filter, for example, if you find, the like only one or two sequence, they don't have any group, you can filter them. The second method, we call it open reference clustering. So here we are using reference, here we are using database. But here we cluster around a reference sequence. That means already we have reference sequences. We download them from there is database for this. There are many reference sequences, database actually, we download them, and then we will align, the reads that we get from the sequencing to these reference sequences. Some of them, they may align, okay, that is fine. Some of them, they might not be aligned. So assume that if you have like a database of 1,000 of reference sequence, and in our sample, we have like 100 different sequence. Some of these, they may align to a specific reference sequence. So that we will take them as, so already we know, we can identify that, as OTU. Okay. But some of them, they might not align to any reference sequence. Maybe there is no reference sequence. Then we will use de novo, like here. In the remaining, the one that they don't have any reference sequence, we will use de novo to create cluster for them. Okay. So that means here we have portion of our sequence will be clustered around a reference sequence, and the other will be, we'll use de novo to cluster them. So here we have hybrid, but also we call it open reference clustering. The third one is closed reference clustering, and here we cluster around the reference sequence. So we have database. We cluster our sequence around reference sequence, and anyone doesn't find a reference sequence to cluster around, it will be removed. It will be removed. We're not going to use it. Okay, so here, if you see all there are pro and con here. This may be more expensive, especially if we have too much data. This one is expensive. That means competition expensive. Okay, so we need more computational resources to do this kind of de novo clustering, and for this it will be easy. For this one, because we discard any sequence doesn't align around or cluster around the reference sequence. So here we lose some sequence, or we lose some information. Again, it's just as up to you. You can take any method of this, or you can use all methods and compare between them. This is something actually So what we get from these three clustering methods, we are getting representative sequence because we cluster around due similarity or reference sequence, but we get what we call representative sequence, and also we call these features. So if we say feature, also we mean representative sequences, okay? And I selected from those OTUs, okay? And we said OTUs are operational taxonomic units, okay? So this is just like show you how clustering, this can take place and so on. And clustering, if you study machine learning, you will know more about the different algorithm you can do that. Again, this is another way of just a diagram that show you how clustering. If you have many sequence, those sequences they can be clustered or can be grouped into a specific group, and after that you take a single representative. Um, you are not taking exact sequence. So this is the difference between AVS and O2U. Here you are taking the sequence that it can represent a cluster. Okay, so this is the feature that we are using it to the downstream analysis. The second method, clustering, the first method. The second method is denoising. And in denoising, here we are getting or we are producing what we call ESV. Okay, it's something or amplicon sequence variance. Comparing ASVs to OTU, ASVs are exact sequence. Okay, OTU, just representative sequence. This is the difference between the ASVs and OTUs. Okay. So again here, denosing, there we were using clustering, and denosing just like new applications or algorithm or programs, they are used to replace clustering. And the idea to give more accurate results. And now most of the people actually they are using these denosing methods, and they are uh, most common programs that are used for algorithm data to deploy and noise. These are the ones that you can use, and each one of these they have um, different way of uh, of creating amplitude sequence variants. But what does denosing mean? From the name itself, actually, it's removing of error because we are expecting um, even if you remove um, if you improve the quality of the read fast you read, it's still are maybe you're expecting there will be some kind of errors coming from the sequencing itself. So these programs they are trying to remove any um, errors that maybe come by chance. The reason why because they need to focus only in the biological sequence. This one. So once you run any one of these program on your read, then you will make sure that the sequence you get is biological generated one. And of course, the analysis, when we do our analysis, we need our analysis to depend on the biological sequence, not sequence coming from any other sources like error or due to PCR or chimeric or any. We need a sequence that we are sure it is biological sequence and it is error-free. Of course, we cannot guarantee that it will be 100% error-free, but at least these algorithms, they try to do their best to give you a sequence, we call it amplicon sequence variance, and it is free of error. Okay. So we have data too. This is the most one that we use. We call it the divisive amplicon denoising algorithm too. And deplore also. The second one also we use it. It uses pairwise Hamming distance of all sequence, and is 3. The design method is used to infer the error-free biological sequence. And this to infer really is very good word because it cannot guarantee 100%. It's just like it's matter of prediction, right? But the prediction is the inference here is really very powerful because tested and it will try to get to the right biological sequence. The biological sequence that when you use it to identify bacteria, then you will feel that okay, so this is the right sequence that we use it for this purpose. So this really, there's algorithm for each one. I loved, to talk about the algorithm of each of this one, but of course there is no time for this. But this is something that you can go and you can go and do, your research on them. But these are implemented in many programs. In many programs, you will find them, you will find out. So now it should be very familiar to you when you data two. Immediately you understand, okay, we are using this for denoising, also, and also deplore and noise. So if you find any one of these terminology in any program in metagenomics, you should understand what they do, okay? Because you may find them in different program. So the step for denoising, and I want you to really to focus on this because when we don't have usually I should give you this in maybe like 2 weeks, to this, and after that we do the pipeline step by step to show you how we do that. Because we are using here a program, we call it Shime, and that Shime actually is a little bit different from other program because you have to understand how to use it. It's not like any other program, but the steps are the same. We need to quality filtering. This is the first step number one. Error correction, then merging parent and read, because from the sequencer, that depends on what kind of sequencing strategy that you follow. It might be parent end or maybe single end. If it is single end, that is fine. If it is parent end, then we need to merge to merge the reads. And by merging read here, because we have forward and reverse read for each one, so we'll bring each read and we will link them like this. We merge them, so we'll make a single read from both of them. Okay, starting from the forward and reverse. All of them, we will merge them like this. So this will be a step. And after that, we move to what we call it dereplication. And dereplication is very important step because this, by dereplication, that we will get rid from any redundant read or sequence. Because we need to focus on, um, we need, at the end, we need to get a single sequence, and that single sequence that we use it to, we use it to, for identification of microbe. So we call it dereplication. And then we have chimera removal, and of course we talked before about chimeric reads. These reads that can be found, aligned to different places. So if it is found in different cluster, for example, that means that it is, you know, this is something that we should get rid of because it will create confusion for us. We need to identify and make sure that our identification to any bacteria is correct. But if you find the same sequence here and there, that means we call this chimeric. We should get rid of it to avoid any kind of confusion. And finally, we'll construct amplicon sequence variants, or ASV, and this is the exact sequence that we will use it. And as I said, when you compare this ASV to OTU, the ASV is more accurate. It's more accurate. So after that, we will come to these slides, and after that, we'll come to the taxonomic assignment, because now we have the features, whether from the whether from the clustering or from denoising, we will get a sequence. So after that, we will identify this sequence, and sequence, there are some strategies that we follow. And by identifying the sequence, we need to taxa, all the taxa, from the kingdom, phylum, class, order, family, genus, and species, and even if strains. So this is what we mean by microbial identification. Okay, so we'll identify all rank. And it depends, because sometimes the software might not be, there might not be sufficient information to identify bacteria. It may stop at any level of this. It might start, kingdom, of course, definitely it's bacteria, but it can phylum, stop here, phylum or class or order or family, genus. If we get to species, that this mean it's really very good resolution. And even if you can go further, like to the strain, also that will give us, more information and so on. So there are two approach, alignment-based classifier, and here we are using, okay, we align to a database. And the second one, we use machine learning classifier. So here we are using two strategy. When we are using database to align the sequence, and this database, we have already known bacteria. So if we align this sequence to database, that means we will identify it just like BLAST. But here we have a database, we call it Green's gene. Or machine learning. Machine learning, of course, machine learning, we predict the class. We are using this machine learning, as we talked before. And if we are familiar, we have scikit-learn and so on. And in machine learning also, there are two approach. We can use pre-fitted classifier. That means already classified, already the model already developed, and you can use it. The second one is you can train your own model, machine learning model, and use it for. on a little bit confused, but we will try to on Monday to cover that because I think two students they will do a project in Amplicon-based metagenomics. So these two lectures are really very important for them. And please try to understand the concept. And in next class also try to understand the steps or the pipelines because they are using that pipelines to do this kind of analysis.